Okay, so good evening, and so my name is Mark Larché, and I am an immunologist, basic scientist, and until I became interested in scleroderma, I'd never really worked on a fibrotic disease before, because the generally held opinion is that fibrosis and inflammation don't go together in some way. Inflammation can be a precursor for fibrosis, but certainly the immune system, I think generally in scleroderma, has not been thought to be particularly important once the disease has been established. So we'll talk a little bit more about that during this talk. So I'm going to talk about some new perspectives in the pathogenesis of the disease, or the cause of the disease, and particularly about biomarkers, what they are and why they might be important, and then we'll just touch on treatment at the end. So you heard from Maggie and Claudia. This is a really tough disease. It's very, very clinically challenging, and it's also very challenging at the basic science end. Maggie showed you that horrific cartoon of all of the factors and cells and pathways that have been implicated in the pathogenesis. The reality is actually that very, very little is really known about the cause of this disease. And you saw in that figure there are so many things that people have looked at, trying to find clues, trying to find a big sort of breakthrough pathway that will allow us to understand the disease and therefore, maybe allow us to target some of the factors, but it remains poorly understood. So I mentioned fibrosis, and scleroderma, until just a few years ago, not sure if the mouse works here, yeah. Scleroderma was thought to be about fibrosis and not and matrix deposition, and not about inflammation. So, I'm an immunologist, I'm really an inflammation guy. And this is one of the early papers that kind of set that ship on course, from a guy in New York, physician in New York who's seeing scleroderma, took biopsies from these patients, put them in culture, and compared them to biopsies from healthy individuals, and basically showed that the fibroblasts that grew out from those skin biopsies in the scleroderma patients were different to the fibroblasts in the healthy controls. And they were different in his hands because they seemed to make lots more connective tissue, lots more collagen. So these are cells taken out of the body, out of the environment that they normally sit in. You put them in culture and they're doing something different. So really that's what set the ball rolling. The sort of obsession in scleroderma research, basic research, with the fibrotic part of the disease. Many people have researched fibroblasts, for example, in scleroderma, looking for a primary defect in fibroblasts, for example. So fibroblasts are the cause. There's something wrong with the fibroblast, but of course fibroblasts can be acted upon by other cells, by other mediators. And that can cause them to have a change in their functional morphology or production of collagen. And we now know that epigenetics can be very important as well in modifying the functions of a cell. So a cell need not necessarily be sort of born with a defect, producing too much collagen, but through the action of the cells around it, the environment that it's in, all kinds of mediators, it can undergo epigenetic changes that turn it into a cell that for the rest of its life produces too much collagen. Okay, so these can be epigenetically determined. So it's not necessarily a primary defect in the cell from a genetic point of view, but more from an epigenetic point of view. So GWAS, this is a study published in Nature Genetics 2010. GWAS, as you probably know, is genome-wide association studies. So this is basically genetic associations. You basically take a large number of people who have a particular disease and a large number of healthy controls, and you analyze SNPs, common SNPs, in their genetic material, and you look for signals that differentiate the disease group from the healthy group. This has been done for pretty much every disease on the planet. Some people are skeptical about GWAS because they say that it doesn't really tell you as much as it probably should, and it's been estimated that GWAS probably only tells you about sort of 5 to 10% of the heritability of a given disease. Probably more than that, maybe 10 to 20%, additionally can come from epigenomic studies, so EWAS, and then the rest, the missing heritability beyond that sort of 30 to 40% is still a bit of a mystery. But GWAS can be important, and this particular study was quite important because it highlighted really for the first time the fact that the genes involved in scleroderma, or that give a signal, the loci, the genetic loci, often seem to be involved with the immune system, either the innate immune system or the adaptive immune system. So this was a bit of a surprise for a lot of people in scleroderma, because as I said, the inflammation in scleroderma is what you see when the patient first presents, but as the disease progresses, the dogma has been that there's not a great deal of inflammation going on, or if there is, it's very sort of low-grade grumbly inflammation in the background. So in this particular study, this is a Manhattan plot on the left, for obvious reasons. Basically, lots and lots of SNPs are evaluated, covering different genes on all these different chromosomes at the bottom. And when you get a locus or loci that are differentially expressed between the disease group and the healthy control group, you get sort of peaks, skyscrapers if you like. And those that fall above this line here, which is a sort of very stringent statistical test to rule out false positives, we consider these genes here to be particularly associated with the disease. Now, like many, if not all autoimmune diseases, the HLA locus or region features very heavily here. So HLA is human leukocyte antigen, which is another name for major histocompatibility complex, and I'll talk about that, the MHC, a little bit later. But the involvement of that region really suggests very strongly that the immune system, particularly T cells, are involved in the disease. And then we have a bunch of other genes here that are involved in the innate immune system, particularly the production of type 1 interferon, which is generally regarded as sort of viral immunity, but can also be involved in fibrosis. And then other molecules like CD247, which again is part of the T cell receptor. So I've shown this over here. This is a signaling chain that forms part of the T cell antigen receptor. So this is the receptor that T cells see antigens with. Those antigens are peptides, as I'll show you in a minute, from self-proteins or exogenous proteins, from viruses, from bugs, from proteins in the world around you. And this chain, the T cell receptor zeta chain, is one of the chains that helps signal to the T cell. So when the T cell recognizes its antigen, that receptor delivers signals into the cell and tells the cell to turn on. And the sum of the strength of that signal is determined by this particular molecule. So you can basically get your head around a scenario where a slight polymorphism in this chain, some changed amino acids in people with scleroderma at the level of the gene, could give rise to a molecule that could signal more strongly to the T cell every time it sees its antigen, or perhaps signal more weakly. It could give you a qualitative change. So that suggests that there's something funny in T cell recognition of antigens in this disease. This is the T cell receptor, and this is what it sees. It's an MHC molecule which has a peptide slotted into the top of the molecule, and here's another view looking down on it. MHC molecules have a peptide binding groove at their surface, and this binds peptides. And as I said, those peptides can be from self molecules or they can be from exogenous molecules. That's how our immune system, particularly T cells, see the antigen. So GWAS in 2010 is telling us that inflammation and immunology is important in scleroderma. This is a sort of more close-up view of that MHC molecule with the peptide inside. And in fact, very recently, the same group led by Maureen Mays down in Texas have really provided more granularity on that HLA association that you saw at the peak of that last Manhattan plot. And this connects this MHC or this HLA observation with the autoantibodies that Maggie talked about in the first part. So these are different types of MHC molecule which look like this. They all have this peptide binding groove, and there you can see a peptide sitting in the groove. Now, this groove is essentially just a bunch of amino acids. Okay? They have charge groups sticking out all over the place. They have bulky side chains, etc. So the peptides that bind in those grooves have to conform to certain charge, size, etc. Limitations. And so the shape of the groove determines what peptides can bind. So from most proteins, there'll be very few, if any, sequences of amino acids that will actually fit that groove and bind with any kind of affinity. Okay, so this is a very specific interaction, but we have lots of different MHC molecules, and some of these polymorphisms at these positions within the peptide binding groove, so amino acid position 96 in the beta chain of HLA-DP, polymorphisms there are linked to the development of anti-topoisomerase antibodies. Okay, so this is associated with diffuse scleroderma, whereas a polymorphism at position 69 in DQ alpha is associated with anti-centromere antibodies. So what this is telling us is that basically there are some peptides, very probably, maybe from self proteins, that are binding to these molecules when they have these polymorphisms, and they're being recognized by T cells in the immune system. And that is leading to the production of autoantibodies. So these are disease-associated genetic polymorphisms. So T cells, we do a little bit of T cell work ourselves. This is a little bit of preliminary data from our own lab, looking at or comparing scleroderma patient cohort with controls. Basically, we stimulate these T cells with self peptides from self proteins of the kind that I just mentioned, and we look for the production of mediators, cytokines and chemokines that are thought to be involved in the disease. So TGF-beta, for example, is a very, very pro-fibrotic cytokine that can be produced by T cells, and in this particular study we found an increased frequency of responses from our scleroderma patient cohort, and the size or the magnitude of production of TGF-beta was also increased. And interestingly, a chemokine, MIP1 alpha, was decreased. So these are T cell related differences, not at the genetic level here, but in the sort of functional biological level. You take the cells out of the patient, you put them in culture, and you can show there are some differences. So again, just providing a little bit more evidence that T cells might be important. Regulatory T cells are a group, a subset of T cells that are important in keeping the lid on inflammatory responses. So you basically have a balance in your immune system of effector cells or pro-inflammatory cells and regulatory cells. The regulators basically suppress the pro-inflammatory cells. And this is an interesting study that suggests that in scleroderma, like many other diseases, actually, chronic inflammatory diseases, there seems to be dysfunction at the level of the regulatory T cell. So although these patients seem to have lots and lots of regulatory T cells, at least cells that look like regulatory T cells, their function is compromised, and that's shown in the graph. This phenotype of cell in the black is a classical sort of regulatory T cell phenotype. These cells suppress very well, you can see in a suppression assay in healthy, when they're taken from healthy controls. This other phenotype is the sort of pro-inflammatory phenotype, and of course they don't suppress well at all. But you can see here in various subsets of scleroderma patients, their regulatory T cells don't function properly. So if you don't have properly functioning regulatory cells, the implication is that you have inflammation that is going on unchecked. So more evidence that immunology is important. Okay, so the next few slides focus on this extremely important paper that came out last year. This is a really groundbreaking publication in the field of scleroderma. And if you're only ever going to read one paper about scleroderma, it should be this one. It's from an international consortium. So Maggie mentioned teamwork at the end of her talk. Scleroderma is a very rare disease, but it's a very good example of where virtually every scleroderma researcher in the world works together as a team. It's really remarkable. I've never seen that in any other disease. People from everywhere pool resources. They're very well organized, and they conducted this study, which is really an exceptionally good study. They decided to look at cells that make type 1 interferon. So I showed you there's a type 1 interferon signal in scleroderma from the GWAS studies. Type 1 interferons are implicated in lots of other autoimmune diseases, including systemic lupus. So they went hunting for cells that produce type 1 interferon. Now, the predominant cell thought to produce that cytokine, or that family of cytokines, is the plasmacytoid dendritic cell. So they fished out plasmacytoid dendritic cells, or PDCs as they're known, from patients with a whole variety of stages of scleroderma, which I can't see without my glasses on, but basically they took them from healthy controls and then from patients with limited scleroderma, as Maggie described, late onset diffuse scleroderma, and then early diffuse scleroderma. And they showed increasing levels of this chemokine here, CXCL4. So in this experiment here, they took the PDCs, they put them in culture, they stimulated them, allowed them to secrete what they secrete into the supernatant. They just took the supernatant then and did proteomic analysis of it, standard biochemistry, and found, in fact, using mass spec, a bunch of peaks that you can see there on the right. And the most abundant protein in the supernatant was this chemokine CXCL4. CTAP3 is the next one, and I'll tell you a little bit about that one later on. And then there are some other molecules there associated with skin barrier function. Here you can see just in the early diffuse scleroderma, the more severe or potentially more severe, expression of CXCL4 in cells in the skin. These are plasmacytoid dendritic cells because when you stain for the dendritic cells and the chemokine, you see dual staining. So this really was the first time that a biomarker, a molecule, had been identified that was linked to scleroderma in the sense that it's highly upregulated, but also is a marker, a biomarker that is able to distinguish between subsets of the disease. So this is a simple ELISA blood test, for example. No doubt it will be transformed into a dipstick test fairly soon. and it can give the physician a tremendous amount of information about the likely trajectory of that patient. So what is CXCL4? To immunologists, it's a chemokine, which means it has chemoattractant activity for cells of the immune system and other cells. But interestingly, to the thrombosis docs, the hematologists, this is a very well-known molecule. It's platelet factor 4, which is very, very important in all kinds of thrombocytopenias. It's a major potentiator of clotting. And it's primarily thought to be synthesized by megakaryocytes, the precursors of platelets, and it makes up about 8 to 10% of the protein content of platelets. And when a platelet becomes activated, it dumps PF4 into the system. And what PF4 does is essentially it mops up all of the anticoagulant stuff that sits in your blood vessels. So it allows blood to clot. So it's a pro-clotting factor. But it turns out that it's also made by plasmacytoid dendritic cells, as this study elegantly shows us, and also monocytes. So this is really why I think this is so important. It's not just a biomarker. You know, sometimes biomarkers are not quite red herrings, but they are markers that are associated with disease. So C-reactive protein is up in everybody that has an inflammatory disease going on, but nobody's really sure whether it's causally involved in the disease or it's just a consequence. This protein, you can see from this list here, has clear functional characteristics that would make you think that it could actually be the causal or a major causal factor in this disease. So it's a very, very potent anti-angiogenic factor. So it blocks the development of new blood vessels. It inhibits the growth of endothelial cells and their division. It inhibits the actions of factors like VEGF, which are known to promote vessel growth, inhibits endothelial cell movement, vascular endothelial cell, downregulates transcription factors that are known to be involved in vessel development and maturation. It inhibits enzymes that degrade CTAP3, which is the other abundant protein in the dendritic cells. And that's a potent fibroblast mitogen, so it makes fibroblasts proliferate. And of course, the fibroblasts give rise to fibrosis. It's chemoattractant for fibroblasts and associated with some forms of liver fibrosis. And then it inhibits antifibrotic cytokines whilst promoting profibrotic cytokines. So I think you can see in that list, this molecule is likely to be very important in the pathogenesis of the disease, which is really, this is a blood vessel, a capillary problem, this disease. The vessels, you get this vasculopathy, which you can see on the nail fold test that Maggie showed you. And this is what causes the tissue injury, the digital ulcers, etc. So the other interesting things they did in this study were to do a little bit more detailed analysis on the clinical outcomes. So basically they did a 2-year study here where they followed people looking for the development of lung fibrosis and the development of pulmonary arterial hypertension, which is shown here, and fibrosis over there. And they stratified these patients into low CXCL4 or high CXCL4. And you can see very clearly that the individuals with high CXCL4, these are all scleroderma patients, did much, much worse. So indicating that this is a good predictor of disease progression, and again, perhaps suggesting that there's causal involvement. So, this is endothelin secretion here. So basically, if you take out blood vessel endothelial cells and you stimulate them with toll-like receptor agonists, these are molecules produced by viruses and bacteria, you get an exaggerated response in these cells. And that exaggerated response is also potentiated by CXCL4. Down at the bottom left, you can see the induction of type 1 interferon, alpha interferon secretion, and this is blocked when you use an antibody in the culture system against CXCL4. In a mouse model, they put a pump into these mice that slowly secreted either PBS in some mice or CXCL4 in other mice. You can see at the top, the normal mice are at the top, they're skin sections, and the mice treated with CXCL4 have dramatically thickened skin, and the brown staining on the right is the infiltration of white blood cells. Okay, and then the skin thickening measurements are given there for you in those other bar charts. Now, a further set of experiments associated with this study looked at the whole range that Maggie talked about. So C in these graphs, C as healthy donors or controls, R as Raynaud's, R plus, Raynaud's patients with autoantibodies, and then very early scleroderma, limited cutaneous and diffuse cutaneous. And you can see that, again, the levels of this particular chemokine very nicely differentiate between these different groups for CXCL4, but not really for any of these other chemokines and factors that have previously been implicated in the disease. I mean, there's a hint there for another chemokine, CXCL10, but it's not as discriminatory as CXCL4. Now, they make the point in this paper that, lots of people have Raynaud's, as Maggie said. 99% of people with Raynaud's don't go on to develop scleroderma, and yet they have elevated levels of this chemokine. And in fact, in those mice that were infused with CXCL4, although they got skin thickening, they did not go on to develop fibrosis. So this molecule isn't the whole story, but it certainly predisposes to the development, and it looks like it's sort of a two-hit or more than two-hit phenomena, where if you have these high levels, you're at risk, and a secondary insult can push you onto the disease. So the conclusions were that the levels of CXCL4 are elevated in patients with various forms of scleroderma, and they're related to lung fibrosis and PAH. What can we do for treatment? Well, this is just an idea, but it's an idea that I hope we'll be able to get going here in Hamilton. The thrombosis guys know that PF4 has a very, very high affinity for heparin. Heparin-induced thrombocytopenia is a disease that is really caused by complexes of heparin, high molecular weight heparin, and PF4. And PF4 binds to heparin because it likes to bind to highly charged molecules. That's part of what it does in the blood vessel. So heparin has been used in the literature to treat Raynaud's phenomenon, really just to try and help with blood flow in digital ulcers. But to my amazement, looking at the literature, it doesn't look like it's ever really been used in a clinical trial in scleroderma, which I find astonishing. So, this is a cheap, widely available product a drug that's been used for many, many years. Scleroderma is clearly a disease that has vasculopathy and micro blood clots at its cause, but nobody seems to have used heparin. So I think this would be a very interesting study to do. Probably the first thing to do would be a pilot study where you actually see, by measuring plasma or serum levels of CXCL4, whether treatment with heparin could bring those elevated levels down. So we can easily do this with the collaboration of our colleagues in thrombosis, just by looking at patients who get put on heparin, if we take blood samples before and afterwards and measure their CXCL4. And then if that turns out to be a good way of regulating the levels of the chemokine, we could go on to do a 2-year trial, just as they did in this paper that I described, to see if it has any effect on the development of the more serious complications. So there are lots of questions remaining, but here are just a few. What induces the dendritic cells to make so much CXCL4? Maybe that's something to do with crosstalk between the T cells. The T cells are doing something funny, as we discovered. Maybe they talk to the PDCs and cause the problem. There is an interesting twist here, which is that there is another sort of polymorphism in CXCL4, which gives rise to a very slightly different protein, which is much, much, much more anti-angiogenic. And I very much doubt that the reagents they used in the study, that I just talked to you about is able to discriminate between those two forms. So that's something else to look at. It may be that we can target this even more precisely. What's up with alpha interferon? I really don't know how that fits into this. These cells also make a lot of alpha interferon, but it's not really clear how that plays into the disease. And then finally, can the vasculopathy, the sort of death of the blood vessels, the capillaries, and the fibrosis that's seen in scleroderma be reversed? Because if you can get a control on the pathogenic mechanism, maybe lowering this chemokine, maybe we would have an opportunity to see whether tissue repair can occur. So it's a challenging disease, involves inflammation and fibrosis. GWAS has implicated the immune system for the first time, and just recently this chemokine has come up as being elevated in scleroderma. It collaborates with the innate immune system to induce type 1 interferon, which is clearly implicated in the disease, but we're not sure how. It's going to be a very valuable biomarker of disease progression and prognosis, and it's an obvious target for intervention. So I'll finish there. You've seen this picture before, and thanks to the Scleroderma Society of Ontario for supporting our research. I'll be happy to take any questions.